Okay, buongiorno. Nice that you're all still here. Last talk before the lunch break, so thanks. I will show you today some data from a recent and still ongoing project that we're running in Salzburg, so that's unpublished data. Mostly it will be brain data, but towards the end of the session there will be also some body signals, and I'm very happy, looking forward to your opinions and thoughts about this. So the project that I'm talking about is about supra-sensory perception, and what do I mean by that? The core research question that we're investigating here is basically whether the conscious perception of hearing a sound, seeing a flash, or feeling a touch, whether it is linked to one common core brain activity pattern or one common body state, as you will see later. And we investigated that with a very simple paradigm. So we have like some chitted pre-trial interval, then the very brief presentation of either a sound, a visual stimulus like a bore, or a very faint vibration to the finger, chitted post-stimulus interval, and then just a very simple response, whether you have perceived something or not. Concurrently, we measured brain activity with MEG. And yeah, as I said, it was a yes-no detection task, and the response mapping for the fingers was randomized over trials. And in the background, that is important, there was psychrometric threshold procedure based on adaptive Bayesian inference that adjusted the loudness of the sounds and the luminance of the gabors and the intensity of the vibrations to reach approximately 50% hits and misses. That worked well, as you can see here. So we had 4 different sounds for reasons that will become apparent on the next slide. The detection rate for all of those sounds was close to 50%. The same was for the 4 different gabors that we presented. And we also stimulated 4 different fingers, from the pinky to the left index finger. Detection was also at around 50% for those. So why did we use 4 different stimuli? That was because we wanted to use a representational similarity analysis, as I will show now, to distinguish 2 different activity patterns in our MEG signal. The one is what we call a sensory model. And the sensory model assumes the highest similarity between one sound and itself, and the highest dissimilarity, for example, between the lowest sound, the lowest tone, to the highest tone. Same for the fingers, from the pinky to the index finger, and so on. So it's emphasizing the sensory similarities between the individual stimuli. And we contrasted this with our supra-sensory model that didn't care about the individual stimuli. And also it didn't care about the different sensory modalities. The only thing that was important for this supra-sensory model was that we assumed the highest similarity within hit trials and within miss trials over all sensory contexts, so vision, audition, or touch, and the highest dissimilarity between hits and misses. Right. What we found, so in terms of correlation between these 2 models and our MEG data, we first found for a sensory model things that we would like to have seen and would have expected. So we found an early peak of our sensory model, and this peak was associated with primary sensory areas, so A1, A2, S1, S2. And for vision, we didn't find V1, but we found cuneus and precuneus regions. By contrast, our supra-sensory model peaked later, and it did so in what is commonly referred to as sensory association areas. So we have a difference in time course and a difference in the brain regions between these 2 models. So what does this actually mean, what we found here? So we found in our supra-sensory model, we found activity that was different for the different percepts, so hits versus misses, but it was similar across the different sensory modalities. And now we looked further into our MEG data. Where do we find this kind of pattern in time frequency resolved manner. So here you see the time frequency maps first for the auditory trials, in the upper row the hit trials, in the lower row the missed trials. And what you can see is this alpha activity relative to pre-trial baseline periods. We have this increased alpha activity, which is higher during sensory processing, and in the hit trials, this then fades away into theta band dynamics. And very differently, we find it for the missed trials. We also find this alpha activity, but it just goes on. And to me, this looks like an internal query, an internal process of was there something, was there nothing? And in case there was something, the participant detected something, this internal process stops. And if it didn't, it just continues on until the response. And importantly, we found the same pattern or a very similar pattern also for the visual modality, and also a bit weaker, but qualitatively very similar also for the somatosensory condition. All right, so we were now interested, as you can see, not only in this difference between hits and misses, but also in this pre-stimulus period. So is there actually something before a sound, a gabor, or a touch was presented that would predict whether you perceive them or not later? So we looked in this pre-stimulus window here. You see that alpha already went up before stimulus onset, and contrasted this between hits and misses across all sensory contexts. We did so now not for across trials average power, but we did it with the so-called burst metric. And so we did the alpha bursts in the pre-stimulus period. And I want to make a little detour about bursts, what they actually are and what they might mean. So what you have seen before was across trials averaged power. And what we often see there is this, what we call sustained oscillations. And many cognitive processes have often been assumed to be sustained or that they are ongoing. But actually, it turned out, if you look into the single trials, this activity is often not so sustained. Often it is like one burst here and followed by a second one. At a different time, there's another burst, or with a different duration on the third trial. So if you would average those three trials together, this would come out. And you would assume that the underlying cognitive process is sustained, when in fact it might be bursty. Another way to depict this, I hope this works, yes it does, is that more graphical depiction. So that would be sustained oscillations, and those would be the bursting oscillations. And these bursts, they are very interesting also from a theoretical point of view. They capture these transient high signal epochs in the data on a single trial level, but they might also be important to choose between different cognitive threats. Instead of like a more sustained activity, you could think about it that it is like more flexible. So it may be used for dynamic and flexible cognitive acts. And they can be modeled by attractor networks. And if you want to read more about this, you can read this in this recent methods paper that I had together with Michael last year or 2 years ago. Right, so we used this burst metric, and we used the bicycle toolbox for that, and we looked at the difference between hits and misses across all sensory contexts in the pre-trial, in the pre-stimulus period, and we found this different. We found more burst events when the participant later on has perceived the stimulus compared to not perceived that. And that was the only metric that was statistically different. There was no difference in the absolute amplitude of the signal, of the alpha signal, in the alpha frequency, and also there was no difference for more intricate methods describing the burst waveform. Importantly, we also tried to relate this to our sensory and our supra-sensory models. So we did this by means of cross-correlation over time. And you can see that the occurrence of those alpha bursts, they predict our sensory model and our supra-sensory model later on, but with different latencies. So what you can see here as like a kind of summary is that we did find such a common core brain activity pattern. We did it with RSA by distinguishing between the sensory model that peaks early in the sensory areas and the supra-sensory model that peaked later in sensory association areas. We also found that those two models were predicted by the occurrence of alpha bursts before stimulus onset. It predicted the sensory model, and then later on the supra-sensory model. So those alpha bursts did not only did predict what you have perceived later on, but also the associated brain activity pattern. Yes, right, so much for the brain data, and now one slide, as I have promised, for body signals. We did not only record MEG, but we also concurrently recorded EOG, cardiac activity, and respiration. And here you see the average over trials. This was our first pass. For all sensory contexts, we found that on HIT trials, the participants make a little blink. So maybe they detach from the external world if they have perceived something and make a little blink. Then we find this shockingly cool anti-phase pattern here between HIT and MIS trials. I have to say, though, that we tried to translate this into circular statistics, and so far we didn't manage to get what we have seen here on the average. So let's wait for that for a second. And then we also found for the respiration data, we found that the participants basically held their breath throughout the trials and then exhaled or inhaled earlier when they have perceived something compared to not. But this is ongoing work. I'm happy to have your opinions about that later on. All right, so that is my final slide. So we have this alpha burst that predict our neural correlates of suprasensory perception, which may be further modulated by ongoing state changes in the body. That leaves me to thank all of you, my team, my collaborators, and my funding. Thanks.
Thanks, Andrea, for the very interesting talk and for the sneak peek. Very much appreciated. Do we have questions for Andreas?
So thank you, super interesting. So with the ECG, is there any way for you to kind of say based on that whether detection was better at systole or diastole, or is it kind of more complicated?
Yes, so this, but
just because it was so briefly.
Yeah, so what it was a bit fast, right? So the orange ones are the MIS trials, and the blue ones are the HIT trials. Mm-hmm. So basically, when at stimulus onset, independent of which stimulus it was, mm-hmm. you were more likely to be in a systole phase and less likely to be in a diastole phase. Mm-hmm. If you have, so more likely in the systole phase when you have missed it, and more likely in the diastole phase if you have hit. This is what this plot mm-hmm. means.
Okay, cool.
All right.
Thank you, great talk. I had a question about this burst of alpha activity. Is it possible, I don't know if it's possible, to locate the origin of this burst in brain? Do you know where are they generated? And what do you think they mean? So is something related to attention, to, I don't know, what's your interpretation?
Yeah, of course. I mean, the concept of attention pops to you into this, but I mean, in this concept, it's also always very loosely defined attention. Of course, when we say it's independent from a specific modality, it is sensory, so it generalizes across senses, so it might be related to attention. That was your second question, right? The first one, where they originate, we are not that far. So we are currently running the source analysis on that. The data that I've showed you was the average across all MEG sensors, just to not have any bias in that. And yeah, we are going on with that.